Greetings to our esteemed panelists. Today, allow me to introduce to you our research entitled Targeted Drug Delivery Systems, the Role of Carbohydrates in Developing Biocompatible Carriers, presented by Group 2, Section B. But before that, allow me to introduce our team, Mr. Japes Clive M. Gonzalez, Miss Rachel I. Pebro, Mr. Ryan B. Shienes, Miss Princess Angie R. Saguban, and I, James Benedict D. Maglinte. So let's proceed to our introduction. Carbohydrates, comprised of carbon, hydrogen, and oxygen, are among the most versatile biomolecules in living systems. Their roles extend far beyond serving as energy substrates. They are also essential constituents of cellular structures and molecular recognition processes. According to Smith et al. On 2021, it emphasizes carbohydrates are classified into monosaccharides, oligosaccharides, and polysaccharides, each category offering unique physiochemical characteristics that can be harnessed for biomedical applications. Polysaccharides such as chitosan, alginate, and dextran are particularly valued for their biocompatibility, biodegradability, and structural flexibility, qualities that make them ideal candidates for constructing drug delivery carriers. And that's according to Johnson and Lee on 2022. These materials are naturally abundant, structurally tunable, and capable of forming hydrogels, nanoparticles, and micelles suited for targeted drug transport. More importantly, carbohydrates play a crucial role in ensuring the compatibility of drug delivery systems with biological environments. Their non-toxic nature and similarity to indigenous biomolecules reduce the risk of immune rejection and adverse reactions upon administration, according to Garcia on 2023. In addition, carbohydrates can be chemically modified to introduce targeting ligands or improve solubility, enhancing the specificity and efficiency of therapeutic delivery, and that's according to Chen and Patel. For example, glycopolymers and mannose modified carriers facilitate receptor-mediated endocytosis, allowing for selective uptake by cells overexpressing carbohydrate binding receptors, and that's according to Nguyen et al. On 2025. This natural affinity to biological surfaces makes carbohydrates uniquely suited for advancing targeted therapy, positioning them at the forefront of biocompatible drug delivery research.
Research gap. Despite the extensive study of carbohydrates in biological processes, their full potential as components of drug delivery system remains underexplored. Martinez and South 2022 highlight that although some progress has been made in using carbohydrate-based materials for drug encapsulation, the mechanisms by which they enhance targeting, specify, and therapeutic bioavailability have not been fully elucidated. Moreover, as noted by Komar et al. 2023, most existing drug delivery vehicles still rely on synthetic polymers that may pose biocompatibility or toxicity concerns. This presents a critical research gap. The need for systematic evaluation of how natural carbohydrate structures can be engineered into safer and more effective drug delivery platforms. So, this section highlights a significant research gap in the field of drug delivery. While carbohydrates are extensively studied in biological process for their full potential as components of drug delivery system remains largely untapped. Although progress has been made in using carbohydrate based materials for drug encapsulation, the exact mechanisms by which these materials enhance targeting, specify, and therapeutic bioavailability are not fully understood. Current drug delivery vehicles predominantly rely on synthetic polymers, which can raise biocompatibility or toxicity concerns. This research gap underscores the need for systematic evaluation of how natural carbohydrate structure can be engineered to create safer and more effective drug delivery platforms. Essentially, there's a significant opportunity to explore and harness the potential of carbohydrates for improved drug delivery, but more research is needed to optimize their use. Research objectives. This research study primary objective is to investigate the role of carbohydrates in creating biocompatible carriers for targeted drug delivery system. The research breaks these objectives down into four specific aims. First is identifying carbohydrates. The study will identify the type of carbohydrates commonly used in drug delivery systems. This involves a literature review and possibly an analysis of existing pharmaceutical products. Second is analyzing biocompatibility and safety. It will analyze how carbohydrates improve the biocompatibility and safety of drug carriers. This likely involves examining the interactions of carbohydrates with biological systems and assessing any potential toxicity. Third is evaluating advantages. The research will evaluate the benefits of using carbohydrates based carriers in tar- targeted drug delivery. This would compare their efficacy and safety to other delivery methods. Fourth is reviewing existing systems. Finally, the study will review the existing pharmaceutical products and research that utilize carbohydrate-based delivery system. This serves the contextualize the research within the current landscape of drug delivery. In essence, this research aims to provide a comprehensive understanding of how carbohydrates can be used effectively and safely to improve drug delivery. The findings could lead to the development of new and improved drug delivery methods. Research analysis. This chapter synthesizes and critically analyzes the finding from the reviewed literature. Without conducting experiments, the discussion will focus on evaluating existing studies to determine the impact of carbohydrates in drug delivery systems. Through comparative analysis, the chapters will explore the advantages of carbohydrate-based carriers, their safety profile, and their application in pharmaceutical formulation. This analysis aims to provide insightful interpretation and highlight future research opportunity based on identified gaps in current knowledge. So, this chapter provides the critical analysis of existing research on the use of carbohydrates in drug delivery systems. It synthesizes findings from reviewed literature without conducting new experiments. The analysis focused on evaluating the impact of carbohydrates, exploring their advantages as carriers, assessing their safety profiles, and examining their applications in pharmaceutical formulations. The goal is to offer insightful interpretations and identify future research opportunities based on the gaps in current knowledge. This approach is valuable for providing a comprehensive overview of current state of research in this area and guiding future studies.
Types of carbohydrates in drug delivery. Identification of commonly used carbohydrates base carriers. Comparative analysis of different carbohydrates types and their suitability for drug delivery. This slide covers the types of carbohydrates used in drug delivery. It will first identify the commonly used carbohydrates-based carriers. Then, it will conduct a comparative analysis of different carbohydrates types and their suitability for drug delivery, assessing which types are most commonly and appropriate for various applications. Biocompatibility and safety of carbohydrates-based carriers. How carbohydrates improve compatibility with biological systems. Mechanisms that minimize toxicity and enhance therapeutic safety. Comparisons with other drug carriers materials, lipids, and polymers. These slides focuses on the biocompatibility and safety of carbohydrates-based drug carriers. It examines how carbohydrates enhance compatibility with biological systems, minimizing toxicity and improving the therapeutic safety. Crucially, it also includes a comparative analysis of carbohydrates-based carriers against other common materials used in drug delivery, such as lipids and polymers, providing a comprehensive overview of their safety profile. Advantages of carbohydrates-based targeted drug delivery. How carbohydrates contribute to improve drug absorption, distribution, and stability. Benefits of controlled drug release in targeted therapy. Limitations and challenges in carbohydrates-based carriers compared to other delivery systems. These slides details the advantages of carbohydrates-based targeted drug delivery. It explores how carbohydrates improve drug absorption, distribution, and stability within the body. The benefits of controlled drug release in targeted therapies. A key advantage of precision medicine are also highlighted. Finally, the slides acknowledges the importance of comparing the limitations and challenges of carbohydrates based against other drug delivery systems to fully understand their potential and drawbacks. Evaluations of existing pharmaceuticals applications. Review of studies utilizing carbohydrates-based drug carriers. Success stories and limitations based on real-world applications. Identified gaps in current research and areas needing further exploration. This, this presentation slide outlines and evaluations of existing pharmaceuticals applications using carbohydrates-based drug carriers. The evaluations involves the three key aspects: a review of existing studies using these carriers, and analysis of real-world successes and limitations of such applications, and an identifications of gaps in current research that requires further investigation. In essence, it's a critical assessment of the current state of carbohydrates-based drug delivery systems, highlighting both their achievements and areas needing improvement.
Okay, so now let's move on to the summary of findings. So first is the types of carbohydrates in a drug delivery. So carbohydrates in a drug delivery shows that research shows that the highlights the potential of the polysaccharides such as the chitosan, alginate, dextran, and the pectin as effective carriers in drug delivery system. So their low toxicity, biodegradability, and modifiability makes them the ideal for the various therapeutic applications, including the targeted cancer treatments, and metabolic therapies. So these carbohydrate polymers also shows the promise in immunotherapy and managing chronic diseases due to their structural versatility and functional diversity. Okay, second is the biocompatibility and safety of carbohydrates-based carriers. So carbohydrate-based carriers are highly compatible with the biological system, breaking down into non-toxic byproducts that minimize long-term accumulations and systemic toxicity. Materials like chitosan, alginate, and hyaluronic acid are well tolerated across various delivery methods, including oral transdermal, and parenteral routes. By enabling the targeted delivery and controlled release, these carriers reduce drug-related side effects and inflammation, making them a safe and effective option for the pharmaceutical applications. So third is the advantages of carbohydrates-based drug delivery system. So carbohydrate based systems offers several key benefits, including the enhanced drug solubility, stability, and the targeted delivery and extended bioviability. So these systems enable precise stimuli responsive delivery, which is particularly valuable in treating complex conditions like diabetes, obesity, and the cancer. By encapsulating drugs within the carbohydrate matrices, dosing frequency is reduced, improving potential compliance and therapeutic outcomes. Furthermore, hybrid systems that combine multiple carbohydrates can optimize drug retention and minimize the side effects, offering a promising approach for effective disease management. Okay, so fourth, or the last one, is the existing pharmaceutical application and studies. So carbohydrates-based carriers have shown promise in treating conditions like diabetes, cancer, and inflammatory disease. Real-world application includes the nasal spray, hydrogels, and nanoparticles that enhance the precision and effectiveness of the drug delivery. Despite encouraging and preclinical results, challenges such as the inconsistent polymer quality, difficulties in loading hydrophobic drugs, and limited progression to clinical trials. Addressing this hurdles through standardization, regulatory compliance, and advanced manufacturing techniques is the key to broader clinical adoption and realizing the full potential of this system. Thank you.
Good day, ma'am. Next is conclusion. This study underscored the vital role of carbohydrate-based carriers in the advancement of targeted drug delivery system. Through the analysis of the existing literature, it became evident that carbohydrates offer a unique combination of biocompatibility, biodegradability, and structural versatility that supports control and site-specific drug release. Polysaccharides such as chitosan, alginate, dextran, and hyaluronic acid emerge as key materials in enhancing drug bioavailability, reducing systemic toxicity, and improving therapeutic outcomes in conditions like cancer, diabetes, and obesity. While numerous experimental and preclinical models have demonstrated the effectiveness of carbohydrates-based systems in real world Applications remain limited by material variability, formulation, complexity, and translational challenges. Despite these limitations, emerging technologies such as stimuli responsive materials, nanofabrication, and computational design present opportunities to overcome existing barriers and accelerate the integration of carbohydrate-based platform into clinical practice. In essence, the incorporation of carbohydrates into drug delivery reflects a convergence of biochemical compatibility and engineering innovation. With continued interdisciplinary research and regulatory refinement, carbohydrates-based careers hold the potential to redefine therapeutic precision, safety, and personalization in modern medicine. The last is recommendations. To build upon the insight drawn from this study, it is essential to translate these findings into practical and actionable steps for research, clinical development, and policy. The following recommendations aim to address identified limitations, strengthen ongoing initiatives, and guide future innovations in carbohydrates-based drug delivery system. So first is advanced standardization protocols. Develop and implement robust methods for the purification, characterization, and quality control of carbohydrate carriers to ensure batch-to-batch consistency, reproducibility, and regulatory acceptance. And the second is invest in scalable formulations strategies. Prioritize the development of cost-effective and industrial viable processes that maintain the structural integrity and biocompatibility of carbohydrates-based delivery systems. Third is integrate digital design tools. Utilize computational modeling, machine learning, and data-driven optimization techniques to predict formulation behavior, enhance drug carrier compatibility, and accelerate development timelines. And the fourth is expand clinical research and trial pipelines. Promote the transition of promising formulation from preclinical models into early phase clinical trials to establish safety, efficacy, and pharmacokinetic profile in human populations. And fifth is explore underutilized sources and hybrid systems to investigate novel carbohydrate sources, including marine and microbial polysaccharides, and explore hybrid combinations with synthetic or lipid-based materials to expand functionality and therapeutic reach. Six is strengthen the interdisciplinary collaboration, foster cross-sector partnership among materials scientists, biomedical engineers, pharmacologists, and regulatory bodies to accelerate the innovation and facilitate smooth clinical translation of carbohydrate-based technologies.
Most, we would like to express our deepest gratitude to God for whose guidance, strength, and wisdom sustain us throughout the entire journey of our research. His presence has been our constant source of hope and perseverance. We are sincerely thankful to our advisor, Mamel, for her insoluble guidance, patience, and encouragement. Her insights and constructive feedback have been instrumental in shaping the direction and quality of our work. To our classmates, thank you for your moral support helpful suggestions, and for being part of an environment that encouraged learning and growth. Your camaraderie and encouragement made this journey more meaningful. We would also like to extend our appreciation to our landlady, whose kindness, understanding, and support during our stay at the boarding house provided us with safe and peaceful space to focus on our research. A special mention also goes to ChatGPT, whose assistance helped us organize our thoughts, clarify concepts, and enhance our writing process. The tool served as a valuable and aid throughout our work. Lastly, to our team members, to my team members, thank you for your dedication, hard work, and cooperation. Each of you brought something unique to the table, and this achievement is a reflection of our collective effort.